Welcome to another Anatomy and Physiology Smart Art video, where we guide you through an important piece of art. After watching this video, you should be able to summarize the process of transcription. Transcription is the process of producing RNA from a DNA template. The term transcription is appropriate because transcribe means to copy or rewrite. Genetic information is encoded in a length of DNA called a gene, which you can see here. The process of transcription rewrites this genetic information into the molecular language of RNA, which is represented here. There are many types of RNA, but here we will focus on the transcription of messenger RNA, or mRNA, which carries the information, the message, needed to synthesize proteins. Let's start by looking at the first step in transcription, gene activation. Gene activation begins once the DNA strands have separated and the enzyme RNA polymerase binds to the control segment or promoter on the first segment of a gene. The strand of DNA used to synthesize the RNA is called the template strand. In the second step, we can see RNA polymerase promoting hydrogen bonding between the nitrogenous bases of the DNA template strand and the complementary RNA nucleotides. The RNA nucleotides are then linked by covalent bonding. Be sure to notice how the DNA and RNA nucleotides are aligning as complementary base pairs. But remember, RNA contains the nucleotide uracil, which binds with adenine. So we see A with T, U with A, and G with C. A group of 3 mRNA nucleotides is called a codon, and each codon codes for a particular amino acid in a protein. Translation ends in the third step, when the DNA template strand reaches a stop signal. The newly formed mRNA strand detaches, as can be seen here, and the DNA strands reassociate as hydrogen bonding occurs between the complementary base pairs. The resulting mRNA strand, however, is still considered immature, so it is in a final process that the mRNA is edited before it leaves the nucleus to direct protein synthesis. Represented here are lengths of mRNA that are non-coding. These are called introns and are snipped out. The remaining coding segments, or exons, are spliced together to create a shorter functional strand of mRNA. To summarize, in transcription, a process that occurs entirely in the nucleus, a length of DNA called a gene is rewritten into the molecular language of RNA, and in this case, messenger RNA. The mature mRNA exits the nucleus and enters the cytoplasm to begin translation, the next step in protein synthesis. We will discuss translation in another Smart Art video. You may ask, so what? Why is it so important for DNA to be transcribed properly into mRNA? The answer is that mutations, small permanent changes in the nucleotide sequence of a gene, can have major impacts on health. Serious disease, such as sickle cell anemia, can result. In this inherited disease, a gene codes incorrectly for a single nucleotide. As a result, the protein hemoglobin is not properly constructed, and the red blood cells are not fully functional. Patients suffer from a host of symptoms, such as breathlessness, severe pain, organ damage, and even premature death.